Welcome back. Today we are going to cover the screening tests of coagulation. I think this is essentially the second most important PowerPoint within coag. The first being just learning the coag proteins, everything that we did in the introduction to hemostasis. This is key because this is what you are actually doing in the lab, the tests that you're performing in the lab. And it's good to understand what does this test mean or what is it testing for? And that will better help you grasp when we get to the diseases and when things are abnormal, why they're abnormal. It would make more sense at that point. So on this side, you're going to see several tests up on the top row and then what they might test for going down. So bleeding time is probably very rarely used, but there are still some sites that do use it. And essentially bleeding time is only testing for platelet function. So you can see here, none of the actual coagulation factors or proteins are involved in a bleeding time test. It's just going to test for platelets. So bleeding time is a test kind of done on the forearm to see how long it takes for those platelets to form a clot. PT, which is prothrombin time, probably one of the most commonly ordered tests in coag, is going to test your extrinsic pathway, which is consisting of 7, 10, 5, 2, 1. I always like to say it in the order they occur. So you can see just the extrinsic pathway here is got the pluses, whereas the APTT, another commonly ordered test, is going to test the intrinsic pathway, which is the bigger of the two pathways. So everything except that 7. Now, you might be thinking, well, I know 13 is involved in both extrinsic and intrinsic pathways because it helps stabilize the clots, the fibrin clot, but it actually does not get tested for by either one of those. And so that's why a lot of times when I list off the factors for the extrinsic or intrinsic, I don't always include 13. 13's definitely got a role. It plays a role in both in stabilizing that clot. But the reason I don't tend to list it in there is because I know that the PT and APTT tests aren't testing for it. So when you go over what does test 13 is a specific test called a urea solubility test. So if you really want to see if somebody might be deficient in factor 13, not having enough, they can order a specific test to screen for that. The TT stands for thrombin time, and this is going to be all about factor 1, which again is fibrinogen. So we're going to get into each of these, but this is just kind of your overall screenshot. All right, so screening tests again are non-specific. They're just going to screen. We're looking to see if there is an abnormality within one of their pathways or within their coagulation system in general. If something comes up abnormal, then we can get to more specialized or confirmatory type testing to detect what truly is happening. So prothrombin time, like I said earlier, very, very common test ordered in the coag world. Again, it's testing for the extrinsic pathway. So that's going to include your common. So again, the factors involved in extrinsic is 7, and then your common 10, 5, 2, 1. This is where knowing those pathways is so key. Knowing what factors are in each pathway really helps you. So it will test for those factors, and basically here's how it kind of does that. It uses a reagent that's called tissue thromboplastin. That thromboplastin is going to mimic the tissue factor, which activates the extrinsic pathway. So if you don't remember any of this, go back to the introduction to hemostasis unit or recording that I did. And in there, it will discuss how the extrinsic pathway starts when there's a tissue injury, and that releases this tissue factor, and that jump starts the whole thing by activating factor 7. Well, in the lab world, we have to mimic that to determine how long it's taking. So they use a thromboplastin reagent that acts like that tissue factor, and then that will activate factor 7, which starts the whole pathway. So the whole time that this analyzer is doing this, it's on a timer because we're wanting to see how long does it take from them to go to starting point to the end point of having a clot. So they use the thromboplastin to act like the tissue factor. We're using incubation, keeping everything nice, warm, and toasty, just like in your body. You have to add calcium back in because remember when you drew blood into the sodium citrate light blue top tube to test. So for those that aren't aware, sodium citrate light blue top is the tube we use for coagulation testing. And remember, we're keeping that blood liquid in there, and so the sodium citrate bound calcium to take it out. so that it won't clot, so that it keeps liquid, well, we have to add the calcium back in. Again, calcium is so essential in clotting. You cannot clot if you don't have calcium. So in order to run this test, we have to add the calcium back in. So it will get added back in here as well with the reagents. And then again, it's all about a timing process. So normally your extrinsic pathway only takes around 11 to 15 seconds to form a clot, which is pretty awesome. So anytime that it is what we call prolonged or longer than the 15 seconds, that is abnormal. That tells us they might be missing something or something might be deficient here, because why is it taking them so long to form a clot? We're not too worried about shortened times. I know I get that question a lot. On my world, I'm not going to worry as much about shortened times. Again, I teach more on a big picture scale here, just overall what happens. So I'm more worried about why does it take longer than 15 seconds for them to clot. So that's typically what we're looking for, because that would indicate something isn't in place in them. All right, the APTT, another common test, is going to test the intrinsic pathway, which also, of course, includes the common pathway. Essentially, it's everything except factor 7, because 7 only belongs in extrinsic. And again, except factor 13, because factor 13 is tested by that urea solubility. So with APTT reagents, we are using an activator substance. So there will be an activator reagent that will help mimic the contact factors. Again, APTT, the intrinsic pathway is activated by contact with some sort of surface or something foreign to it. And so we're going to mimic that with an activator. We're also adding a phospholipid substitute for calcium to help make sure we're forming a clot. And again, it's a measured process, a timing process. So because this is a longer pathway to go through, intrinsic has a lot more steps to it, it's going to be a little bit longer of a time frame. So normally it's around 23 to 36 seconds for someone to form a clot in the intrinsic pathway. So again, we're looking for prolonged times. Anything greater than 36 might give us a clue something is happening. And then the thrombin time. This is a test to all about fibrinogen, which is factor 1. So it is actually measuring the conversion of fibrinogen to that fibrin clot. That is at the end of both pathways. And so we're adding thrombin reagent to the patient's plasma. And again, it's a measuring process. So anytime you get a prolonged thrombin time, it's going to indicate something with their fibrinogen. So it's either they don't have enough factor 1, they are deficient in fibrinogen factor 1, or they have dysfibrinogenemia, which essentially means they have the fibrinogen, but it's not behaving the way it should. It's dysfunctional. I always think of the dys for dysfunctional. It's just not acting like it should, so it's not going to form that clot like it was supposed to. The other reason that you would have a prolonged thrombin time is if the patient is on something that interferes with the testing. A really good example here is heparin. If somebody is on heparin therapy, this will automatically prolong their thrombin time as well. It inhibits that process. All right, there is a test called the Reptilase time. This is using the venom of Bothrops atrax, so it is that's why it's called Reptilase, from that reptile. So again, same purpose here as thrombin time. They are looking for the conversion of fibrinogen to fibrin. The only deal that's different between thrombin time to Reptilase is that Reptilase is not affected by heparin therapy. So if you know somebody is on heparin therapy and you wanted to measure their fibrinogen conversion, it'd be better to do it with the Reptilase time because it will not be sensitive to that. Again, otherwise you would see the Reptilase time prolonged for the other same reasons, deficiency in fibrinogen or dysfibrinogenemia. The only one that's different is it just is not sensitive to the effects of heparin. Okay, so let's go through a few little interpretations. So if you have a prothrombin time that's abnormal, meaning it's prolonged, and an APTT normal, what factor do you think is most likely abnormal here? So if you know your pathways, you will know that the only thing that is not in the intrinsic APTT world is factor 7. 7 only belongs to prothrombin time. So it is only factor 7 then that could be abnormal here. The common pathway, again, 10, 5, 2, and 1 are found in both. So it would have to have both affected if it was something in the common. So because it's only the prothrombin time that became abnormal and the APTT stayed normal, it's factor 7 that's probably the most affected. So here's that example. If you have both abnormal, then it's one of the common pathways factors. So 10, 5, 2, or 1, because those are found in both intrinsic and extrinsic pathways. So if you have a normal prothrombin time and an abnormal APTT, then it's one of the ones that only belong in the intrinsic. So that would be 12, 11, 9, or 8. So it's one of those. And you could probably say pre-K, HMWK, but I don't talk a lot about prekallikrein or HMWK. They are definitely part of the contact factors. They're part of the extrinsic, but they're not really getting tested by the APTT. That's not something we're too worried about. It's more 12, 11, 9, or 8. Okay, so once we see an abnormal screening test, we again will do confirmatory testing where we're going to kind of figure out, okay, what's exactly the problem. So a lot of times you can just do factor assays where you're actually measuring the factor levels that are present in that person. And if you see a lower level, that will tell you, well, they're deficient in that factor. You can also do what we call substitution studies, or commonly called mixing studies. Now, these aren't done as much, but I know that there are definitely still labs out there that do these. So it's something that we still discuss, but a lot of labs aren't performing these as much anymore. And then again, if you think it's something specifically with factor 13, we could do the urea solubility test to specifically screen for that level and function of factor 13. So we'll start with the mixing studies. These get a little bit tricky or complicated for people to get. I personally love them because I feel like they're like a puzzle. So once you get them, they're a little bit easier. It's just initially understanding them. So what a substitution or mixing study involves is you're taking the patient's plasma and you're mixing it with a substance that we know exactly what's in that substance. So some of the substances we're known to use is normal plasma, serum, adsorbed plasma, or aged plasma. And so we know exactly what factors belong in those. And by mixing it together and determining the results, we can kind of rule in, rule out what factors is causing the issue. So once we mix the substance with the patient's plasma, we're going to repeat the PT and APTT tests and determine if it helped it. Did it make it come back within reference range? If it helped it, that means whatever was in that mixing study gave back what the patient was missing. So if that patient was deficient in, say, factor 10, and we mixed it with something that had factor 10, it should correct their test. It should make it back to normal. So again, it says here, if abnormal test is corrected by the mixture, the component has replaced the deficient factor that was missing from the patient's plasma. All right, so the key is knowing what's in every single thing. So normal plasma, it's going to contain all the coagulation factors. So this is used a lot to detect different inhibitors that can circulate in somebody's plasma that will inhibit the testing in the analyzer. So there are different like lupus antibodies, things like that, that can inhibit the testing process. So this is just kind of used to screen for that. Serum, and this is where it gets to be kind of pure memorization. Unfortunately, I know that's never a great thing to hear, but it is memorization. So serum is going to lack the factors 1, 5, 8, 13, and 2. I know I listed those weird, but basically you need to memorize what it does have, and it has 7, 9, 10, 11, 12. So I always remember it has 7, S and S go together, serum 7, and then it's basically 9 through 12. So that's what it does have in serum. Aged plasma has everything except 5 and 8. So it has 1, 2, 7, 9, 10, 11, 12. So it's easier for me to remember that it's just missing 5 and 8, has all the others. And then adsorbed plasma lacks the prothrombin group. Last week, or in the introduction to hemostasis, which I did last week, that's why I was going to say last week, but in that unit, we discussed the prothrombin group, and that's made of factors 2, 7, 9, and 10. It is missing this group, so it has the rest, 1, 5, 8, 11, 12. So for me, again, adsorbed, I just memorize that it's missing the prothrombin group, and so I know it's the other ones that it does have. All right, so there's your chart on what test results would happen in the PT, APTT, and what you would see with the different mixtures and what would be factor deficiency. This seems ultra confusing. Basically, it's just going to take a lot of example problems to kind of figure this out and get there. All right, so let's do a few review questions. If a patient has a prolonged PT but a normal PTT, what is the most likely factor deficiency? So this is one that we discussed earlier, and that should be factor 7. Again, factor 7 only belongs in the extrinsic pathway, which is measured by PT. So if only PT was prolonged, then your factor 7 is the one that's most likely deficient. D-dimer. Now, we did not mention D-dimer in this PowerPoint, but it is definitely something that you should be aware of. A D-dimer we mentioned in the introduction to hemostasis unit. Basically, a D-dimer is testing for clotting. It's testing for if somebody has a clot present. It's commonly a test done in the ER or clinic situations in which the patient is presenting with all the symptoms of potentially having a clot. So they could run a D-dimer test to kind of screen to get a yay or nay on that. And so remember, your clotting pathway, the fibrinolysis pathway from the introduction to hemostasis unit, discussed how it was plasmin to plasminogen to degrading the clot. So to degrading the fibrin clot. So I'm sorry, I should have said, yeah, plasminogen to plasmin to degrading the fibrin clot. So it should be the third answer here. So we're not degrading fibrinogen. Fibrinogen turned into a fibrin clot. So it's number 3 there listed, or letter C, however you want to say it. Which of the following factors is not present in adsorbed plasma? So this again is going back to knowing what's in adsorbed. So it's missing 2, 7, 9, 10. So which of the following is not present? 2, 7, 9, or 10? Well, there is number 2 right there. So that would be 2. The integrity of the intrinsic system is evaluated by what? So intrinsic system is evaluated by APTT. If a patient has a prolonged PT and prolonged APTT, but both are corrected by aged plasma and serum, but not corrected with adsorbed plasma, what is the most likely factor deficiency? So this is one of those mixing study problems. So I'm going to take it off the slideshow format because I want to write on here. So the very first thing that I do when trying to solve this is first look at my PT and APTT results. So if both are prolonged, that tells me it has to be something in the common pathway, because it needs to be in the common in order to be prolonging both. So that is choices 10, 5, 2, or 1. And you'll see that's our choices right here in the multiple choice question. We already rolled it down to that anyway. So now that we know it's one of those possible 4 factors, let's look at the mixing studies results. So it said corrected by aged. Let's remember what aged plasma has. It has everything except 5 and 8. So it has 1, 2, 7, 9, 10, 11, 12. So as you can see, it has everything except 5 and 8. So if it corrected it, that means it has to be one of the factor it did have. It gave it back and made it work. So it can't be anything that it didn't have that's missing. So you could rule out factor 5 here as a choice, because 5 is not in here, so it would never have fixed it if it was missing 5. So we can rule out factor 5. We know it's not that factor. All right, serum also corrected it, and serum has 7, 9, 10, 11, 12. So it has all of those, so it corrected it, so it had to be one of those. So we can rule out anything that wasn't in serum. Well, 2 is not in serum, so it couldn't have been that to correct it, and 1's not in serum, so it couldn't have been that either to correct it. So the only choice left is 10. 10 is the most likely factor deficiency. But just for the sake of learning, let's go through the very last one anyway. Adsorbed plasma remember, has everything except the prothrombin group. So it has 1, 5, 8, 11, 12. Because remember, it's missing 2, 7, 9, and 10. So if it has these ones and it did not correct, that means it absolutely could not give back any of these. So it can't be any of those. It has to be one of the ones so it was missing. So this still makes sense as factor 10 because it didn't have factor 10 here and it did not correct. So it couldn't have fixed it. So that is the answer, is factor 10. These are confusing and it's normal for people to not understand them immediately. It usually just takes time of practicing these kind of things. Okay, going to the next question. The urea solubility test is specific for detecting deficiencies of what factor? And that again should be factor 13. An abnormal thrombin time is associated with what? Again, thrombin time is all about measuring the conversion of fibrinogen to fibrin. So it's all about fibrinogen. And so if it was abnormal, you're probably deficient in fibrinogen out of these choices. And that's it. The last slide is just my references. These are two amazing hematology books. I love both of them. There are, of course, other wonderful hematology books out of there. These are the two I'm more familiar with. So that is it. It does get to be a little tricky, like I've said before. Coag is a small little department in the lab, but man, it is a tricky little sucker. So just keep practicing those mixing study questions and just knowing really what's in your intrinsic and extrinsic pathways is really going to help you. Have a wonderful day, and we will talk again soon.